Hello, welcome to the meeting. No one, I don't think anyone's going to show up today, maybe a couple people later, but I'll get started for those of you on YouTube, and you can follow along. I can provide you with the links in our Slack, and if you need an invite to our Slack, just contact me, and I can give you the link into the Slack. So let's get started. Today we have a number of things going on. We have the last day of GSoC, and I know that Mainak is probably busy submitting his final submission because I think the deadline is today or tomorrow. Good luck to Mainak, and hopefully he gets everything in without incident. He just sent me a message before the meeting kind of going over everything. His last GSoC pull request, which was accepted into our GitHub repository. So let me go to that now.
Yeah, Bradley, can you hear me?
Hello, how are you?
Just joined.
Yeah.
Yeah, I'm doing good. Hi.
Hi. Let's see. Okay, there we go. How are you?
Yeah, I'm doing well.
Good.
Yeah, so I saw you were actually showing the report that I had built, right?
Yeah, I was looking it over. Yeah.
So
did you want to review it with us, or do you want to?
Yeah, actually I've got a presentation to show, so maybe I can go ahead.
Okay, go ahead.
Yeah, okay, then I'll share the screen.
All right.
Just let me know if you can see.
Yeah.
Yeah, can you see the screen?
Yeah, I can see it.
Okay, now I'll... Okay, so this was the report that you were showing, right?
Yeah.
Okay, so I actually... See, this report is actually very long and it might be a bit tedious to read. So what I did is that I converted that into a short presentation. So I'll just go over it quickly. So, okay, so this presentation is actually about the GSoC project for this year, which was upgrading DevoLearn. So I'll just get started with it. Okay, so the first thing that, the first model that I actually had to build from scratch was actually the model that could segment the nuclei from the C. elegans embryo imagery data. This was actually sourced from the Cell Tracking Challenge website, and the dataset essentially contains 3D plus time fluorescence microscopy data of the developing C. elegans embryo, and it also contains the corresponding segmentation maps as shown to the right. And like here in this image, you can see that the photos to the left, they are basically the microscopy imagery data, and the images to the right, they are segmentation maps, which are there in the data. So the dataset was by no means ideal, and it was scattered across many folders. And I actually had to do a lot of cleaning up because in the extreme points of the z-axis or the dorsal ventral axis, there were a lot of data points which were not really of any use because they are empty. So I had to clean them out, and then the images are also stored in a very strange format, and which was actually not known to me at that point. So that actually added to the challenge. So I'll just go ahead to the next slide. Okay, now coming to the training part, the model that I had used is basically the architecture is basically of a feature pyramid network, and like maybe you could read the FPN paper. There is a very famous paper which basically laid the foundation for this sort of architecture, and the backbone is basically a ResNet 18. So I have used a library called Optuna to find the learning rate and the batch size for training, and I have used those parameters for the training, and I had actually trained the model for 50 epochs, and the results are and the training metrics are shown in this image that you can see. Is the image clear, Bradley?
Yeah, I can see it.
Yeah. So as you can see in the image, in the validation lies loss, it goes down as we train, and the learning rates also are decaying because I opted for a decaying learning rate to make sure it doesn't get stuck anywhere. So okay, now coming to the model performance. So these are a couple of examples that I basically tested out the model on. So to the GIF that you can see, it shows inputs which are varying over z-axis, and the segmentation map that is coming out of the training model is shown to the right. And this image to the right, it shows some inputs from the validation dataset which were being used for testing essentially. And like in the first column, you can see the input images that was fed into the model. The second column shows the original mask that was present in the dataset, and the third column shows the prediction that the model was able to make after training for 50 or so reports. So they seem pretty close as of now, and I think the results are pretty much acceptable. So okay. So I'll just go to the next slide. Okay. So this section deals with the upgradation of the cell membrane segmentation model. So this was a model that was basically already present in the DevoLearn library, and the first thing that I looked at when I started out this expedition for upgrading this thing, the first thing that I looked at was the data that was being used for training the older model. And the data, what I saw is that the data was essentially in a .mat format, and they were being converted to a .jpg format. So, which is actually leading to a lot of loss near the segmentation map edges. So, the code, I saw in the code that, so this issue, it was basically tackled using an OpenCV based operation, which had led to larger gaps between the segmentation maps, as you can see in the left part of this image. And so I had to refactor the pre-processing part entirely, and I had to save it as a .png file, which led to a far better segmentation map data that we can see to the right. So this was the thing that I fixed in the data. So go ahead. Now coming to the training part, here too I used a feature pyramid network as is the same architecture as the last segmentation model I talked about. Okay, so now here also the case was very similar. I had used Optima and I had run about 100 or so trials, and which were run on only a little fraction of the available data for one epoch each to make sure that the trials are fast and quick. So the metrics are shown to the right. Now I'll go ahead and show you a couple of things after training. Okay, now to the left, I have compared the models, the new one and the old one, with a lot of noisy and like underexposed data. So the image to the top, this input image, as you can see, there's a lot of noise in the image, and the old model, it sort of performs well in this case, but the upgraded model, it performs way better than the old one, because the segmentation map is actually a lot closer to the original mask that was provided. And the same goes with the other one. As you can see, the exposure of this image is very, very low, and the older model completely fails to map anything. But the upgraded model sort of has a better idea of what's going on, and the results that it provides is very close to the original mask, except for a couple of flaws. So this part is essentially to show the difference, the improvement that has happened. And to the right, we can see this GIF which shows the stages of the cell, and of course the older model outputs are shown to the left, and the upgraded model outputs are shown to the right. And as you can see, the segmentation maps are more tighter, I would say, and the gaps between the cells are very low. So I think that's where the main improvement is happening. It will be able to map the final parts of the cell a lot better. So coming to the next section. Okay, so this section deals with the deployment of the DevoLearn models. So the thought behind this deployment of the DevoLearn models to the web is basically to basically make it way more accessible than it is, because installing a Python library in a laptop or in a computer might be a bit difficult for people who are not having a lot of experience with their computers. So I figured it would be a better idea to have some sort of a GUI for the usage of these models. So the features, so essentially the basic features are, it has a drag and drop input function, It also contains example inputs in case the user doesn't have any inputs written at that point. And there's also an option to zoom into and then pan and view the images in a full screen mode. So this basically shows the two main branches of the DevoLearn project that we are at right now. Because up to this part, up to third stage, the saving models part, everything is the same. But then I would say the software has basically merged into, it has basically diverged into two parts, which is the PyPy package and the online web app. I'll be talking about the web app in the following slides. So, okay, so this is the web app. Okay, I'll just go ahead and open up the web app then. Actually online.
So this is live now?
Yeah, it is live and I'll just load up the. Okay, so until the web app loads up, I'll just go ahead and show you a couple of screenshots from the web app. Yeah. So these are the screenshots of the web app. So to the top left, we can see this image, which is showing the home page of the web app, and the rest of the images they are showing the various functionalities of the web app that has been deployed online. So I'll just go ahead and check. Okay, it's taking a bit of time to load. I think it's because of my network, maybe. Okay, so let's wait for a while until this goes up. Or maybe I should try it in a comment. Okay, it's loading. Yeah, can you see the screen? Can you see it?
Yes.
Hello?
Yeah, I can see it.
Yeah, so you can see this is the home page, and I've linked to the datasets, and a couple of more links are here. And plus, I've also linked to the GitHub and the PyPy packaging in case the user wants to install it. In case the user actually finds purpose from the online demos, maybe it could run more inference using the library.
Yeah.
So this drop-down menu to the left, it will lead to the three of the models that has been deployed online, and the user has the freedom to select any of these models, and he'll be able to run inference. And okay, so this image that you can see here, this image is, it's actually one of the examples that are present in the web app itself. So I'll just go ahead and change the example. Okay, so the example has changed. I'll just press run, and yeah. So these are the results that we're getting from the web app, and the backend is basically the same neural network which is present in the PyPy package. So in the backend, the neural networks are running, and in the front end, the user gets the segmentation maps and the centroid map. So I'll just go ahead and do the same thing with the other example. Let's go ahead with example 1. Okay, so this image, it seems very, very underexposed. So let's see how the model performs on this. So this is the that we're getting as the segmentation map. So the edges are very crisp and clear. So yeah, that's the output that we're getting here. And in case the user wants a full screen, in case he wants to check it out in full screen, he has the option to do that, and then he can zoom into it, and you can actually zoom into it and see how the model did. Let's just go ahead and show you guys the third example, the third one. Okay, I'll just go ahead and press run. Let's see. Okay, so this model, it basically predicts the lineage populations from an image. So we are getting the lineage names here and the predicted populations to the right. So this is the current status of the DevoLearn web app, and I'll just go ahead and open the presentation now.
Okay. That looks very good, yeah.
Yeah, so these are the screenshots that I was talking about. And okay, now coming to the threshold slider. I guess it would be better if I show you the example live on the web app because it's open right now. Just show you what the threshold slider does. I'll go ahead and go to one of the segmentation models. So you can see the screen, right? You can see the yeah. Okay, so this threshold slider basically applies a threshold to the model output. So the model it basically outputs an image which contains pixels, and these pixels, the values of these pixels, they vary from 0 to 1. And this variation it basically corresponds to the confidence of the model in in that certain pixel. So if I set the threshold to be very low, to be a very low value, 0.01, which is very close to 0. Let's see what the outputs are. Okay. So in this output, as you can see, the edges are a lot more closer, and in some cases there are a couple of overlaps. Especially in this part, there are a couple of overlaps which might be favorable in some scenarios. So what the user can go ahead and do is that he can set the threshold to be a very high number. This is 0.9. If you press a run, you'll get a result which on first glance it might look very similar to the other result, but if we zoom into it, there are no overlaps here, and the overlaps are essentially eliminated in this sense. So this could be a useful feature for the users who are looking for a more fine-tuned experience. So the edges are coming close, but they are not really overlapping, which could be important for the user. And from what I could infer, like in case the user wants a more accurate centroid mapping apart, like if he is designing a more accurate centroid map, then he could turn down the threshold, and then a larger part of the cell would be covered by the segmentation map, and the centroid mapping would be more accurate in that case. So that's one of the outcomes of this threshold slider. And the same goes with the other model too. Let's go ahead and give you a quick glimpse of what it can do. So if I set the threshold to 0, let's see what happens. If the threshold is set to 0, like it gives out a very scratchy image, which is not really useful by any means. So I'll just go ahead and set it to some low value. Okay. Yeah. So as you can see in the segmentation map, there is a little bit of white spot in this. But if I set the threshold to a high value, let's say 0.64. So that point is now gone. So the output it's basically less sensitive to the finer details in this case. So in case the user wants a cleaner output with less stray points, then he might go for a high threshold, and in case he wants a bit more details, and if he's willing to risk some stray points, then he might as well set the threshold to be low, and he'll get these extra points which the model is trying to gather. So yeah, that was the thresholds, which I've also added screenshots in this presentation in case the model doesn't work in the last moment. But it worked. So
hey.
Hello, Krishna.
Yeah. Okay, so yeah, thanks.
Yeah, I'm sorry to interrupt.
Yeah, it's okay.
Okay.
So as I was showing.
Okay, I have to say something. Can you just
Yeah. Go ahead.
So, Bradley,
Yeah.
I joined the meeting, but now I have some work to do. Can I leave now?
Yeah, go ahead.
I'll come back.
Okay.
And by the way, let me share something.
Can you wait until Mainak gets finished, or?
Yeah, I'm just posting it.
It's just the last slide.
Okay.
It's essentially done.
Yeah.
Okay, so I was talking about the web app object. So this GIF, it basically shows the interactive part of the plots, which I already shown in the live demo of the web app. So I don't think explaining this is required, because I showed you the zoom part and the full screen part and the panning, all this part, and I've also explained the threshold slider part. So I think this slide is not really required here, but it's still there. So yeah, in case the model doesn't work in the last minute, as a backup. So that brings me to the end of this presentation. So yeah, thank you.
Thank you. Yeah, the interactive plot was really nice. I mean, that's something I haven't seen before too much in this type of thing, but I think it's useful. People like to visually explore their data, so I think that helps.
Yeah, it does help. It does help. To look at the finer details and to see if the model really works in their kind of data. So I think this would be useful.
Yeah. Well, thank you very much for that. And when are you submitting your project to the portal?
I'm submitting very soon, within a day or two.
Okay.
at most.
When is the deadline? Do you remember when the deadline is?
Yeah, it's the time of, yeah.
Okay, so yeah, just make sure you meet the deadline, and the time is, you know, in like I think UTC, or I don't know what it is, but make sure you know the actual time because they cut it off. So that's good. All right. So thank you. Hello, Akshay. How are you?
Hello. Just joined.
Yeah. So
are there any more questions? Then I'll just stop sharing the screen then.
okay. Well, I guess we can come back to it in a little bit. If Krishna wanted to share something, did you want to share something, Krishna?
Yeah, sure. And by the way, nice work, Mainak. It was really awesome.
Yeah.
And hi, Bradley. How are you?
All right. How are you?
These weeks are so tiring. I did a neuro match.
Oh yeah.
So, he wanted to present a paper, but then I suddenly got a call. Actually, they're trying to help a family to, get relocated from Afghanistan.
Oh yeah.
They are acquaintance of Dr. Richard Gordon.
Okay.
So we are trying that either we could get them refugee status in India or Canada. So I got called to that. So I'll be trying to make it. So I also made a presentation that I'll share in the chat. So it's a paper. Can you be to my screen?
Are you sharing your screen? Not yet.
Okay. Can you see now?
Yeah, I can see.
Okay, so really good paper, and it's regarding finding the cell after division from machine learning, and when this thing was done manually, a lot of assumptions were to be made regarding everything that would make them mostly very inefficient because you can't predict everything. Some of your assumption always don't fit right. So now what they do that they make a probability distributor function, and they use that probability density functions. So what they do, they take the cell size at birth and division condition of their histories. So with that, the trained probability density function reproduce and confirm the previous result and pre-assume assumption of the size control and show that the cell division is indeed stochastic. So what does this mean? That this process happens by iterations, and every iteration of the probability density function improve itself and see that it is like fitting right to the data that we have. And they use two open source data here. Okay, so if you people go through the paper, you can see that data here, and it's freely available. And it has single cell measurement. And what else? Okay, so real quick, these are some of the diagrams that even I don't understand fully. Maybe they are too biological. So if I just have to, if I have to go to the computer science stuff, then it's really awesome models, how they are using it. And since they are making it iterations by iterations, maybe if we could also incorporate our own cell division data into this model, we can even further improve it, because these are they use two data sets, combine them, and most probably we will be also having some cell division data. And if we could merge our data into theirs and then rerun the training algorithm, then it might give even better results. So what else? I don't know what this thing means, to be honest, because I don't have much of a biological experience. But okay, there's one more thing. Bradley, I want to ask you, what is this curve?
What is what? This plot?
This curve.
It looks like they're modeling the probability density function in terms of different parameters.
So, have a question, like, is there any SI unit for cell division? Like divisions per second or something like that?
Well, yeah, I mean, like people will measure divisions, like, so it's usually a function of the lineage tree. So when there's a branch in the lineage tree, there's this, division, and you can time the divisions out. And the timing is not always uniform, it has some variation in the interval between divisions.
Does it consider temperature also, because different temperature would have different divisions?
It can, yeah. It can. The cell's metabolism can vary with temperature, and so you have, yeah.
Okay, so like we take some default temperature. For example, we have 2 bacteria and see which one divides faster. So we will took a standard temperature for checking the divisibility.
Okay.
I'm asking this, do we take a standard temperature for that?
Well, you can usually people set that for, depending on the cell or the organism, there's usually a standard temperature. It looks like down here, if you go to the legend of this figure, E. coli was cultured at 37 degrees Celsius, sometimes 25 degrees Celsius. That's usually like a baseline temperature. And then, you can vary the temperature, and that can introduce like heat shock or some sort of other effect on the, on the cell division.
Yeah. So what they are doing here, like mathematically, neural network sense, they are taking the current parameters, and they're also using the previous parameters output, like they are forming a recurrent neural network here. So division that would be happening now would also depends upon the division that has happened a little before. So that is also and it is a very interesting paper. I read it and I didn't understand it fully because a lot of things were so biological. I didn't get to know what is happening to be exactly. So I would really like to discuss this paper in more detail, and I've shared that in chat. So,
okay. Well, let's see. Let me see if I can open it up here, and I'll share my screen to show we can go through it a little bit if you want.
Yeah, sure.
Hello, Jesse. I think that's Jesse. Yeah.
Hi, Jesse. How are you?
So this is can you see my screen here?
Yes.
Okay, so this is the paper, and it looks like they have, so they're using a neural network. So what they're doing, and we've done this sort of before, kind of modeling lineage trees and that, but they want to understand like how cell size differs by division. So sometimes when cells divide, they usually are about half the size of their mother cells. And then, there's this, they can grow in size a little bit, or there's an asymmetry in division. So if you take a mother cell and you divide it into 2, each daughter cell will be half the size of its mother cell. But sometimes there's an asymmetry in that size. So sometimes maybe one daughter cell is like 60% of the mother cell, one other cell is 40%. And so that depending on the organism that holds, or there's some sort of scaling between the two. So this is where they take a neural network and they try to predict like cell size control, which is actually something that the cell does as it matures and as it divides. And so you get the mother cell, and then the daughter cells emerge, and sometimes the daughter cells expand a bit in terms of their size, and then this goes on and you end up with an organism that's much bigger than the embryo. So you get this, but you have to, there's this, as cells divide, there's this sort of control that controls the size of the cells and ultimately the size of the organ that it's going to form. So there's different non-size control mechanisms that actually depend on history. So it says there's a conditional dependence on the size distributions of history. We had a discussion about this in our group on Saturday. a rather lengthy discussion about like the role of historical contingency and neural networks. And so, you know, or Markov models, actually, where Markov models don't have a history. And so there are ways at which you can like incorporate a history into your Markov model, which is basically where you're transitioning between states. And so we had a discussion on that, but it looks like that's what they're doing here as well. In this case, it's a neural network, but they're actually trying to incorporate a historical aspect to it. So it's drawing from previous data, you know, maybe like the last 20 or 30 examples, and it's kind of using that as not as like just a training, like a general training model, but like as a time dependence. So it's a bit different because the data, instead of just sampling generally speaking, it's, you know, there's this time, it's picking up this pattern in time. And as you know, things that happen in time are temporally dependent. So, you know, your state at one point in time correlates with your next point, you know, your next state in time, but over long distances, it may not correlate at all. So there's this dependence on history that's important to, you know, understand if you want to look at a dynamical system, for example, how they, you know, how that behaves. And so they actually used E. coli, which is a bacteria, and they used S. pombe, which is a yeast. And those are single-cell organisms, so they're a little bit different than C. elegans in that they're single-cell organisms that, you know, they divide and they form colonies. And so they have the size, you know, they have the size that they, you know, when they divide, they have a size. They can't get any bigger than something. They can't get any smaller than something. And in single-cell organisms like this is important in terms of like metabolism and so forth. So they don't use like a metabolic model. They don't use an internal model. It looks like they use this sort of model of, they kind of, you know, assume certain things about the size. They just use the scaling law, which is where you have this trend in the data, and then you can get a range of outcomes. And it's a stochastic generation, which means it's sort of drawing from a distribution at random. So, you know, again, you have these distributions, like a Gaussian distribution is just drawing from that. So that's, yeah.
A question?
Yeah.
So, like all single cells follow similar lineage tree, like somewhat similar lineage tree.
Well, I mean, lineage tree just basically, let me show you. So, you know, the lineage tree looks something like this. Let me get a pen here. So, you know, usually it's like this where you have a branching pattern. And then let's say that this lineage tree is asymmetrical. So this mother cell here will divide at a certain time. And then you'll have two daughter cells. Those daughter cells, though, will divide at different times.
Yeah.
And then that's... So you can see that they don't always divide at the same time. Sometimes they divide synchronously, but sometimes it doesn't. And there's, you know, there are different reasons for this. It could be in a multicellular organism. It could be that they're going to form different organs. Sometimes if... Yeah.
So like the daughter and mother cells don't need to have like similar time frame for division.
No, they don't. They don't. Sometimes, like in C. elegans, there's a sort of a programmed time for this. So, like, you know, if this lineage is going to go off and form some, you know, organ that needs to be formed in a certain amount of time, then that's going to be, you know, something that is, you know, it's going to divide at a certain time scale. This thing might be the germ cell, so it divides at a different time scale. I mean, so there are differences in terms of the organ. Now with like the organisms they're talking about, yeasts and bacteria, I don't think there's any sort of like a strong asymmetry like this. It's just that they divide stochastically over time, which means it's just like, say, there's a Gaussian distribution with a mean of like 10 minutes. This is just a hypothetical because it's not biologically plausible, but then it would just sample around that 10-minute interval. Maybe like anywhere from 8 to 12 minutes, you get to have another round of division. And so this just happens, over, that's a very slow, it just will draw over that time. There's no real pattern to it, but it could be asymmetrical that way. So that's what they're trying to model. They're trying to model like, different scenarios for divisions, some asymmetries, but it's not going to look so much like this. In C. elegans, say, you might get this asymmetry, and it might be due to like the formation of organs. But in this case, it's totally stochastic, which means it's just drawing at different times. So this is their microfluidic devices figure, and this is like something that they use to constrain the cells. It's like a flow chamber for cells. They put them in there, and they use the flow chamber to sort of measure a division. So the mother cell is coming through here, and then there's a flow a shear force that pushes the daughter cells out, I think. And then there's this birth size and generation time. So you can see what I was talking about before, what I think Krishna was talking about, where you start at a size, and then the cell grows, and then it divides. And so then it goes back to a sort of a characteristic division size, and then it grows again, and it goes back. So, this mother cell is going to start off like this, and it's going to grow to this size, maybe twice the size of its division size. And then it'll divide, and the daughter cells will be this same size as the mother cell was when it divided. And you just keep going over and over, and you get this step function where over time you get this cell. It expands, it divides, it expands, it divides. And you can imagine if you have a colony, that the size of that colony would expand, I think, roughly twofold, because you have every round of division, you have this doubling of the size, just by virtue of the cells growing and dividing like that. Now, so they can observe that in microfluidics. And then I think this stuff here just shows kind of, so they're using the medians of the conditional probability density function, which is just like a function, like a Gaussian distribution, where you have a mean and then you have tails, and they're using the median because sometimes, in distributions there's skew, and there are other things that make the mean impractical to work with, so they use a median. And we don't really know what biology is doing, other than just having this drawing from, and I mean, they're not necessarily drawing from a probability function. They're just drawing from, like, their local neighborhood. Like if the division time, like I said, is 10 minutes, it could be like, either side of that. The timing isn't precise, but it's generally within that window. And then temperature will affect this time. So if you have a lower temperature, your time will be longer because the cell's metabolism is lower. If the temperature rises, the cell metabolism can be shorter, plus you have other things going on with increased metabolism. So RL equals reinforcement learning. Oh, this is a thing here. Akshay asked a question. Markov decision processes have this state-action pair, also used commonly for RL, where they use an algorithm like Bellman's equation to predict the most optimal future state. Yeah.
Okay.
So that's good. Yeah. Thanks for that.
And thanks for explaining the paper.
Yeah. I think that's good. I don't think there's a lot more, but it's just we've done some stuff like this in the past. We've done a lot of stuff with like lineage trees and looking across organisms. And it's interesting, like different organisms will have different, like sometimes they'll have different symmetries.
Yeah.
So like instead of like a twofold symmetry like humans have or C. elegans has, like in marine marine invertebrates, they'll have like 8-fold symmetry. So like the organism is the same around this circular axis. So it's a weird world there, but
and the paper was quite interesting, and maybe in future we can I'll go through it, and maybe if I don't get something, maybe in future we can discuss more.
Yeah, that would be good. Thank you for that, Krishna.
Do you have like some division data on C. elegans division?
Yeah, we do actually. We have, I think, we have a number of data sets, and they're in the CSV format, and they have a lot of division times and subdivision. So, like, we've sampled the division times and gotten derivative division times and all that. I'll have to pull that up and put that in the Slack.
Yeah, if possible, please share that to me, and maybe we could like do something to it.
Yeah, that would be great. Yeah, and so yeah. All right. Any other questions before I move on? So I wanted to talk about this repository I found on GitHub, and it's this repository on PyGRN. So we've talked about GRNs before, and this is something that they're called genetic regulatory networks. And so you can guess that this is going to be doing these in Python. So this is forked from D9W, who actually is someone who we submitted a paper to this workshop last summer, and it was he was one of the organizers of it. This was at the A-Life conference, and so they're interested in computational development, developmental biology, and then agents, developmental agents. So they're doing this project PyGRN, which is where you can model these artificial gene regulatory networks. So a gene regulatory network is where you have a model of a gene. So it's like something that, like a binary sequence that encodes something like a phenotype. So you could create a quote-unquote gene for maybe like a pencil, all right? You could describe a pencil using a binary string or a series of binary strings. So you could have like multiple genes describing a pencil. And that pencil is going to have, it's not something that is uniform across all pencils. There are different features in a pencil that vary. So like I have a pen, a digital pen here, which differs from the pencil I have on my desk in a number of ways. And so how would you generalize that pencil to these different models? The answer is that you would build a gene regulatory network, which means you take each gene and you have something called a promoter at the front. And the promoter is a conditional device that allows you to activate it at a certain strength or activate certain parts of the gene as needed by your problem. And so in nature we can observe regulatory elements that regulate genes and their expression. So sometimes genes are expressed, at a very high level, sometimes at a very low level. Sometimes certain parts of the genes are expressed and not others, given the environmental context. And so and then of course you can use this to model objects in the real world, like pencils or building car bodies or whatever you want. You can use this as a modeling exercise to build things and to regulate the shape and the details of something based on a simple model. So, if you had a car module that said, I want to generate shapes of a car, cars don't all look the same. There are different details, but they basically have the same sort of body plan, as it were. And so you would model that body plan using the genes, the genetic encoding, and then you'd have these regulatory elements that will allow you to sculpt it. And so this is what this program does, or the software does. And so these are the instructions here. Usage, testing. I don't know if they have a good README in terms of like showing what it can do. They just have the setup and all this. But I wanted to bring this to people's attention, that this exists, because this is maybe something that's useful to people. If you're interested in, say, genetic algorithms, or you're interested in generative modeling, this is a good way to go. And we haven't really talked too much, well, we haven't really gotten too much into like how these artificial models work, but you know, they're going to be, you can use a lot of computational tricks to get it to work and work in different ways. So people have been experimenting with these gene regulatory network models in like at conferences like Gecko or Artificial Life, and you know, but it's something that isn't like really widespread. So this is something that you might want to check out. Another thing I want to share is that we have our Google Summer of Code stub on the website. So this is the website for DevoWorm, and let me put it in the chat. And our Summer of Code stub has our people who are the students in every year, the students who are in the program and accepted into the program. So this is Mainak here, class of 2021. Mainak, if you have a better picture of yourself, send it to me. But I just took a screen grab off of one of our sessions. We have, last year we had Mainak Deb and Ujjwal Singh, who did Open Devo cell integration and then the beginnings of DevoLearn. So, and Ujjwal also did something, sort of a sub project under that called DevoWorm AI. So if you weren't here last year, there's this DevoWorm AI repository which has a bunch of models. So, you know, at this point we have a lot of AI models for looking at cells and analyzing cells, cell shape, cell size, and things like that. So this is like a nice collection of models, and maybe this fall we'll kind of reevaluate where we are with that and what we can use it for, because we have all these tools. We haven't really used them for a lot yet, and we need to maybe use it a little bit more. So then the year before we had this Open Devo Cell project by Vinay Verma, and that was a nice semantic segmentation package. Arnab was the year before, and he did this developmental networks and XML frameworks for the C. elegans embryo. This is actually where we looked at some of the timing data, or we refined that data a bit. So we were doing that a while back. And then in 2017, Siddharth Yadav did image processing with ImageJ. So he actually created an ImageJ module as well. So we have all these tools, and I don't know exactly where we're going with this. It's something that we'll need to sort of focus on how to implement it, you know, in a way that's, you know, maybe we can create some sort of nice analyses from this. But there's a way, we really have a lot of we have a lot of tools in place now. I think that's going to be an exciting time coming up for the group. And then go to the submissions document, and there isn't much here new. I'll reiterate this NeurIPS workshops list. So if you're interested in submitting something to a NeurIPS workshop, the schedule is now online. And what you do is you go find one of these sessions and you try to find it on your favorite search engine, and you can maybe find their website where they will tell you what the deadline is for submission. There isn't a lot like at the intersection of biology and AI, but there is stuff on like more technical issues, more technical topics in machine learning and in reinforcement learning and so forth. The other thing, we had this mathematics at DevoWorm. We talked about that last week. That's still ongoing. Like I showed a couple of additions to that. I know Jesse didn't see it last time, but we can go over that maybe at some point. I can put that go over that in more detail on another meeting. And so we have we have the Kindle book and the Boring Billion. So the Kindle book is something that Krishna was working on. I know he's here now, so I wanted to bring it up. And we have, yeah.
So yeah, I've not like worked on it, and I think that it won't be available in anywhere like near future.
Okay.
It is like at least like 2 or 3 months away till now, and all the members like have to discuss a lot and brainstorm a little bit. So what to put in between. I already have made a cover page and what would be the chapters be, a list of chapters, but still I need everyone in the group group needs to be in the session so we can, discuss further.
Oh yeah, I think so.
Yeah. And I need approval of everyone to finalize the chapters.
Yeah.
And maybe like next week when I'm free with the neuro match, we can certainly do that.
Yeah.
Yeah, sure.
Yeah, that's good. I mean, I'm glad you're coming along on that. So yeah, we'll review that next week. Let me go back to sharing my screen. So yeah, I think that's it for all the things that we're going to talk about. And I want to get into the paper's final thing here. Let's see, I think this is it here. Okay. So I have a couple things to talk about. I think this will be interesting. Let's see. So first of all, there's this interesting thing I found. There's this guy, David Kipping, who runs this YouTube channel called Cool Worlds. He runs a lab called Cool Worlds, and it's actually not biology. It's cosmology and astrophysics. And he has this idea about, like, he talks about the Earth in terms of a very long history. So he's interested in this question of, like, what happens, sort of, how does the world end? So if you're not aware, the world will end eventually in terms of, like, our solar system. So the sun has a finite lifespan. The sun, as a star, will at some point run out of fuel or get close to running out of fuel. It'll expand in size and heat up, and Earth will be obliterated. Probably the life will die off before that because it'll get pretty hot, in terms of, like, the sun being getting hotter and brighter and larger, and then it'll actually engulf Earth. And so that means that our sort of any solar system, in fact, not just Earth, any solar system where there's life, there's this finite window for life to exist. And so if life starts at a certain point after a planet is formed, then, how long can life sort of sit on a planet and evolve? And, do we get something like a technological spacefaring civilization to emerge? So the question is around 600 million years ago, the first animals evolved on Earth, such as Dickinsonia. If we were to rerun the tape from the starting point, do you think a technological spacefaring civilization would reemerge on Earth before our planet becomes inhospitable? And people said no by a small majority. And he says, I don't have a good answer for this because the start time above is somewhat arbitrary and causally connected to prior history. Okay. So this brings up an interesting question, and that is that people have looked at this topic in the past, and there's this old question about replaying the tape of life. And so people have revisited this question periodically, and they've asked, if you start from, like, the emergence of life, if you replay that tape, does life look similar to what it looks like today? In other words, if I just went back and I just replayed the tape of life, forgetting all we know about, like, life on Earth, and this could be true of another planet where there's life, would you have primates, for example? Or would you have dinosaurs? Or anything, would you have life on land? Or maybe life would look a lot different. Maybe it wouldn't even be anything that resembles life on Earth now, or life on that other planet. And so the question is basically you're replaying this process of evolution and recombination and all of this and reproduction, and you're getting this result. So people have done this experiment, but they've never really done it with an endpoint in mind, which I think is interesting, that there's this time window for life. And people have talked about, like, if you start off with a planet that's hospitable to life, but the conditions aren't right, it may not give life a long time to sort of intelligent life a long time to emerge. And so these are all questions you can ask by replaying the tape of life. And so this actually came from Stephen Jay Gould about 30 years ago. He used this metaphor of replaying life's tape. And so this is Gould, I believe, thought that, like, everything would be dramatically different. Every time you rerun the tape, things are different. And so we don't really know, but this relies on the sort of stochasticity of the evolutionary process. So that means that if you rerun the tape of life, things really can't be the same because life is stochastic, and you have enough events, stochastic events, that you end up in a much different place, or it's very likely to end up in a much different place. And so a direct approach to quantifying evolutionary predictability requires massively parallel, carefully controlled, and fully characterized evolution experiments. And so people have done this experimental evolution with bacteria, with yeast, with other organisms, C. elegans. Usually, if you have a short generation time, you can do large-scale evolution experiments, where you can do maybe 1,000 to 5,000, even like 100,000 generations over a number of years. And so you can get a lot of data on what life, at least in these organisms, will look like if you replay that tape. And so, you get different mutants, but this isn't really the question. The question is that, can you start off and then end up with this great complexity? And so people have done this sort of work, and this is a review of this area of work. But people aren't really close to answering it. It's still one of these things that's kind of a mystery. And so this paper, Replaying the Tape of Life in the 21st Century, kind of updates this idea a bit. This is where, first of all, we need to have an experimental evolution where we can ensure this sort of replicability, because then we can't say, if we don't have that, we can't say whether, whether it's just a statistical artifact or whether stochasticity is overcoming this repeatability. But accumulating data is now challenging this classic answer of no. So we have this experimental issue, and if we can overcome that, we can really get a good answer. But then, you can look at other things other than experimental evolution. So you can look at a survey of alleles described in the literature that cause non-deleterious phenotypic differences among animals, plants, and yeasts. That indicates a similar phenotype can often evolve in distinct taxa. They're independent mutations in the same genes. So you sometimes have convergent evolution of things like wings, or actually flight or wings, where they have this functional thing, but it's done in many different ways. So like insect wings and bat wings, those things are very different in their structure, but they both achieve flying or flight, and they both have sort of a similar structure. So these things sort of speak to, maybe if you play the Tape of Life again, maybe you do end up with more or less the same outcome. So, we have to take these sorts of things into account as well. Imagining other possible paths for evolution runs into four important issues. One is resolving the influence of contingency. So this is, if history gives you a certain hand, you have to play that hand. You can't get another hand from the dealer. And so that always has an influence on things. Imagining living organisms that are different from the ones we know. So, maybe there are different phenotypes that emerge very early, and so that makes a difference, whether that happens or whether there's some constraint there. Third is finding the relevant concepts for predicting evolution. So how do we even know how to predict evolution? And if we understand those principles, maybe we can understand this problem better. And for estimating the probability of occurrence for complex evolutionary events. So, if we understand that multicellularity is this fluke or the stochastic thing, then, we understand that maybe it can't happen twice, or maybe it happens in a very different way. But if there are certain conditions that need to be met for multicellularity, and they're actually quite common, then, we have this possibility to have a repeatable evolution across planets or whatever. So this is an interesting question. It kind of goes very deeply into a very traditional area of biology. And there's a lot that we can, people have done these studies again and again. So, if we wanted to make a contribution here, it would be very much you'd have to find a niche area. But nevertheless, I think there's some interesting things to say here. So, extrapolating from short bouts of evolution to the entire span of life evolution. So a lot of our experimental evolution is based on these short observations. So there are things that, you can focus on and try to understand a little bit better. And finally, this goes back to even the wall of complexity that Stephen Jay Gould talked about, which is that, you know, when you have something like bacteria, which are very simple, and in the Precambrian dominated the biota on Earth, and they still dominate the biota on Earth, by the way, but like they were the only game in town back then. There was a lot of complexity in that type of organism, you know. But then as you go farther out in terms of complexity, there's less and less frequency of occurrence, and there's less and less complexity in specific lineages. So this is this wall of complexity where you get these bacteria that have a large number of constituents, and you get fewer constituents as you go through time. So there are fewer opportunities to really make very large-scale changes. Plus, you have this historical contingency working against you. So these are all things that you need to think about with this. So I mean, that's an open-ended question.
Yeah, that's really cool to me, as you probably know, Bradley. Yeah. But could you share that paper somewhere specifically? Because I'm really interested in that. Yeah,
I can share some more detail about that. I think.
Did you happen to see in the other Slack there was a paper that Johan posted that was about I heard this guy actually from a researcher, but really quickly I'm going to mention really interesting paper called "What Behavioral Abilities Emerged at Key Milestones in Human Brain Evolution: 13 Hypotheses of 600 million year phylogenetic history of human intelligence." That combined with the paper that you just showed were kind of at some point I'm going to do some kind of a specific project about these topics because reasons and you know I like them so.
Yeah, that would be great. Yeah, I saw that paper, and that's an interesting. This is where they're putting behavioral traits on a phylogeny or a tree, and they're looking at how the sort of the order of their emergence. So,
I haven't gotten deep into that paper yet, but just I really like the perspectives of trying to expand our view on not just one generation, but big time, holistic. Species level existences and things like that. So this is, I'm just saying, stating my interest in that. So if you can let me know where the paper could be found.
Okay. Yeah, I'll put them in the Slack, in the OpenWorm Slack. So I'm going to go through one more paper. I did want to mention that I was planning on doing this liquid crystal biology, so this was for Susan's benefit. I don't want to do it because she's out she's getting surgery, I think. But I did want to point to this book, The Physics of Liquid Crystals. So this is something that I put in this OpenWorm Slack, and this is something that it's an old book from 1974, but it really opened up not only a lot of technology, like if you're familiar with liquid crystal televisions, but also in biology, where people are looking at bacterial colonies or other types of multicellular colonies, and they're looking at how they're organized. And so liquid crystals have this property where they have an alignment amongst the particles, or in bacteria, it could be bacterial rods. And you get these different phenomena like jamming phase transitions, where if you get enough of them in one space, they stop moving. Usually bacteria will kind of flow around, or they'll move around. If they get jammed up, there are too many of them in one place, they get jammed up. And so that's a phase transition in terms of their organization across cells. And so there are a lot of interesting things in this book. I put it because it's a classic book. People might be interested in reading it for reasons other than I think we talked about a paper that featured some of these topics last week, and so I'm going to come back to this when Susan's back, but I wanted to because I put it in the Slack, I wanted people to understand why that was. So that's an interesting classic book. Mainly I'm going to go into this paper, I think. So this is from MIT Tech Review. This is it's an AI paper, it's machine learning, and it's an endlessly challenging playground teaches AIs how to multitask. So we talked about this actually on again the other meeting on Saturday, where we talked about virtual worlds and sort of how they provide a nonstop stream of open-ended challenges and nudge AI towards general intelligence. So DeepMind and a couple other groups have created these virtual environments where you put agents into them and you let them explore the space. And this serves. We talked about environment a couple weeks ago. We had the series on environment and how environment can influence the organism and development. And so this is one example where if you have a behaving agent or even a developing agent, just where the cells are dividing and being influenced, you can have a virtual environment in a model of the agent, and then a virtual environment that really specifies the environmental conditions. So in this case, you have an environment where there's this maze, there's geometry, and the agent is developing, or it's behaving. It could be developing, maybe not, and it's interacting with these geometric shapes, such that it's maybe telling it where things are in space, so you can encode different landmarks, or in the case where you have to learn what a step is, you would make a step up from one layer to another, and this agent can learn what a step is or how to generalize to other types of steps, or a tower, what a tower is, how to climb a tower. So there's all these things that the agent can learn in an environment like this. It couldn't learn if you're just programming it or teaching it from pictures or something like that. So this is they can build these 3D environments, put agents in them, and then the 3D environment. And a 3D environment is limiting because, like I said, with embryos, you can have things like temperature, which really influence the state of the agent. So if you have a developing agent that's a developing group of cells, you might have a temperature chamber here with some geometry to it, and other like light sources that come in and out of play. It would be a little bit more abstract than this, but you would have this environment where you could control all the parameters and then test different values for these parameters to see what happens. So this is very exciting because the technology is coming about where you can actually put agents in a 3D world and you can actually train them, and they've used this mostly for reinforcement learning. So these playgrounds, the agents will use trial and error to try to figure out how to manipulate or how to interact with this environment. And so again, this is much better than if you had like a bunch of pictures that you train the agent on, where you just hard-coded something into the agent. And so the playground manager has no specific goal, but the general idea is to improve the capability of the player over time. So, there are actually platforms where you can download the platform and you can actually just put agents into it, because these cost a lot to make, a lot of time and expertise. And so if you have these pre-made environments, it's a lot easier to just pick them up and start training things in it. So this is having learned to experiment, these bots improvised a ramp. So here they learn that you can use some feature of the environment as a ramp, and so that's what they're doing. It's not necessarily obvious to the programmer maybe what that is, but that's what the agents are learning for. So, yeah, this is from the MIT Tech Review. So they have, they have these AI generating algorithms, AIGAs, which are genetic algorithms which generate AIs, so they're algorithms that generate AIs, and it's an interesting area here. This is something that they're working on in OpenAI, but there's a lot you can do with this. I think it's very, very flexible. So I'd be interested in hearing, if people were inspired by that, maybe we could have another discussion about like how these environments, how we could use them, and I don't know. It's just a really exciting area, I think.
Yeah, will that be also in the stuff that you share on the Slack?
Yeah, I'll share that as well.
I'm interested in that too.
Yeah.
That's good, thank you.
You're welcome. All right, yeah, there's Mainak. And so Mainak, good luck with your submission. And if you have any questions, let me know. But I think it's pretty straightforward.
Okay, sure.
And I looked through your submission, and everything looks to be there. I don't think there's anything missing.
Yeah.
Okay. All right. Well, thanks for attending, and I'll be putting that information in the Slack. And next week, we'll see if you have anything you want to present, like Krishna did this week. Let me know, and I'll make time for it, and we'll follow up. I know next week it'll be officially the end of GSoC, so we'll probably talk about some other things, but maybe we'll have a discussion about how to integrate a lot of our tools into one place, or that paper that we have been promising on DevoLearn. So, all right, everyone, have a good week. Talk to you later.
Yeah, you too.
Thank you. You too. Bye.